5 ish. So if we start now, we will be finishing 11:25, 11:30. So straight now, go to the first speaker. Jane Clear is a lecturer in epigenetics at the Institute of Developmental Sciences at the University of Southampton in UK. And she has been working for a long time in fundamental mechanisms of placental function. Is doing a very nice analysis using trophoblast cells, analyzing epigenetics and placental tissue from the Southampton pregnancy cohort. So, Jane, thank you very much for being here today, and the screen is all yours.
Thank you. Can you see my screen okay?
Yes, everything.
So thank you for this opportunity to actually talk about some of the work from the Southampton Placenta Lab. We're interested in how epigenetics may be a mechanism that links the exposures experienced by the placenta to how it functions, how it supports fetal development, and how it may program the risk of the individual developing diseases in later life. So we're interested in how factors in the maternal environment, such as body composition, hormones, nutrients, even drugs and toxins and possibly pollutants, impact upon the developing fetus, and how this leads to the programming of lifelong diseases, particularly the cardiometabolic disorders, including hypertension and obesity. So we know that sort of the environment perceived by the mum will influence how the baby develops in utero, and that will stay with it throughout its life course. So the original epidemiological studies highlighted that actually the smaller babies are the ones that are associated with this increased risk of disorders such as hypertension, type 2 diabetes, and obesity. But further studies looking at data from the Dutch hunger winter actually started to sort of establish some of the mechanisms underlying these observations. So they saw that depending on when the women underwent this exposure to famine, there were different effects on the weight of the fetus. So not all of the babies had effects on birth weight, but there were still effects on the offspring in terms of its cardiovascular disease risk and effects on its metabolism. So what was happening was there were stable changes either to tissues or to gene expression profiles within these individuals, which were sort of programming or transmitting the in utero environment through the lifespan of the individual, leading to these disorders in later life. So what we're interested in is really understanding what is programming these stable changes to structure and to gene expression in response to the maternal environment. And that's something I'll come on to. So what mechanisms are underlying these changes? But actually, how can we identify those individuals that have been exposed to different environments in utero and may have been programmed to this increased risk of lifelong cardiometabolic disorders? So we're interested in studying the placenta because this is the interface between the fetus and the maternal environment, and so is perceiving all of these cues from the maternal environment and maybe changing its structural function to support the fetus and lead to changes to its development and its risk of lifelong health effects. So the placenta is really important in terms of having the appropriate structure and function to support the fetus throughout pregnancy to achieve its appropriate developmental status at birth. So in terms of the human placenta, I just thought I'd show you an image here of the human placenta so you can understand what its actual structure is and how really important it is that this placenta develops appropriately throughout pregnancy so that it can support the developing fetus. So this is just a cartoon image of the placenta, and what we can see is that it's actually made up of these tree-like structures. So this villous tree shown here at the top and down here and there. And so if it doesn't have the appropriate surface area of this villous tree, the placenta is not going to function to its full capacity to support fetal development. So this image here is just a close-up showing one of these villi. So we've got the fetal blood on the inside, we've got the maternal blood on the outside, and we've got this yellow layer here, which is the syncytial trophoblast. So actually, it's this syncytial trophoblast that's receiving effects in the maternal environment and carrying out all the functions to support the developing fetus. So in my diagrams, I'll just show it here as the syncytial trophoblast in yellow. So it's important that this, as the functioning unit, can carry out all the functions of the placenta, so transporting nutrients, for example, across from the maternal blood to the fetus, and transporting waste away from the fetus, and providing a barrier to protect the fetus. So we're interested in studying how the placenta works in terms of its structure and function, but what underlies this is actually the gene expression. So the placenta must have the appropriate genes switched on in the right cells at the right time during development, so that it can carry out all of these functions to support the fetus. And the thing that is regulating this gene expression is epigenetics. So epigenetics will be determining which genes are switched on at the right time in the right tissues. And all of this together will make sure the placenta is able to support fetal development appropriately. But the placenta will be having signals from the maternal environment. So all of these factors will be influenced by factors in the maternal environment, such as nutrition, hormones, toxins, etc. And these are going to adapt. So we will be able to monitor the placenta in terms of how it adapts to things it perceives during gestation. But also these changes to the maternal environment are going to determine how it functions in terms of protecting and responding to further insults during pregnancy, and how it functions to transport nutrients, etc., across to the developing baby. So we feel in the placenta lab, it's really important to understand what's going on in the placenta at all of these levels. So what do I mean by epigenetics? So epigenetics is basically the factor which determines which genes are switched on or off at the right time in a tissue or at a specific stage in development. So they determine whether the gene is transcribed and goes on to become protein. So by these regulatory processes, we're generally referring to histone modifications, so things that control the chromatin structure. So basically how tightly packed the chromatin and the DNA is, so that the transcriptional machinery, such as the RNA polymerase, can either not get into the DNA, or it can get into the gene, as shown here, and it can switch on gene transcription. The main epigenetic process that is studied is DNA methylation. And generally we're studying methylation, so a methyl group shown here in red attached to a cytosine, which is in a pair with a guanine. And these are normally clustered in what is termed as CPG islands. So these clusters of CPGs are normally present in the gene regulatory elements. And generally, these will be unmethylated so that the transcriptional machinery can come in and it can switch on transcription of the genes. So it's like a switch that determines, okay, it's open, you can take the machinery in and you can switch on transcription. Whereas in some cases, these areas have to be methylated because we want genes to be switched off. So no transcription is occurring. And so this is the way we control whether genes are switched on or off. And this is important, particularly in imprinted genes or in X inactivation, or in certain situations, we may have faulty methylation, maybe in cancer or due to environmental influences, where we are turning off genes that we don't necessarily want switched off. So these methyl groups attached to gene regulatory regions are really important for determining whether things are switched on. So I'm mainly going to talk about DNA methylation when I'm referring to epigenetic changes, as this is generally the most studied process at the moment in placenta. So DNA methylation is really important for conferring the cell-specific differences in gene expression and therefore phenotype, for the developmental differences in transcription. So actually it's really important from the very first stages from fertilization and the developing embryo for establishing the placenta and then the branching and the development of the placenta to form the appropriate surface area in terms of that villi I mentioned, so that it can carry out the appropriate functions. It's also really important for genomic imprinting. So genes that are imprinted in a parent of origin manner so that they are switched on or off for the trait associated with that parent, which I will talk about in a minute. So DNA methylation is really important for these three factors, but if the methylation is disturbed, this can have great effects on the individual. So it's involved in the establishment of many human diseases. So it underlies a lot of cancers in terms of the inappropriate growth and differentiation of cells, also growth defects in terms of fetal growth restriction. And what we're suggesting is that it may underlie some of the cardiometabolic disorders that are being programmed in utero by changes to fetal development in response to changes in the placental structure and function. So we're suggesting the placenta is really important in terms of its DNA methylation, and is also really prone to programming of changes to DNA methylation, because during very early development, there are changes to methylation which will affect the placenta. So following fertilization, there is global demethylation, and then there is remethylation. And at this stage, we have the blastocyst, where we have the perfectoderm cells forming, which form the placenta. So these have a lower DNA methylation status. So the placenta generally has a state of low DNA methylation, but it also has a high number of imprinted genes. So these are genes that withstand the fluctuations of and changes in methylation throughout development. And these are really important in terms of growth restriction or growth promoting effects. So the placenta and the fetus can be affected by changes to epigenetics in a number of ways. We've got these processes that occur naturally with changes to methylation. And so there can be epigenetic drift in terms of the methylation patterns when we have the changes to these throughout development. We also have the impact of imprinted genes, so they're really important for development, and changes to the methylation status to those can occur. And then we also got environmental factors that are going to impact upon the DNA methylation of globally and of specific gene regions within the placenta and the fetus. So we know that factors in the maternal environment, such as smoking or gestational diabetes or obesity, are associated with changes to the DNA methylation in the placenta. We also know that when we look at cohorts of placentas from small for gestational age babies or large for gestational age babies, they do have altered DNA methylation profiles. So what we want to understand is what is the mechanistic linking changes to the DNA methylation in response to the environment to the phenotype of the individual, because we know a lot of these DNA methylation changes are going to be important for fetal development and therefore its subsequent risk of disease. And so one example is the imprinted genes. So we look at these in the placenta because these are regulated by DNA methylation, and they are really important for regulating fetal growth. So the placenta has a high number of these, and the paternal expression of these is to promote fetal growth, whereas the maternal expression is to decrease fetal growth. And this normally is in balance. But when this goes wrong in terms of altered methylation, this will lead to alterations in the placental function and how it can support fetal growth, and therefore alterations to fetal growth. So in these imprinted disorders, so changes to the methylation of imprinted genes leads to either fetal growth restriction, small for gestational age, or large for gestational age fetuses. And particularly with these are extreme cases of small or large for gestational age fetuses. You see the cardiometabolic phenotype developing in the individual as they grow. So we're interested in how these changes to methylation may impact upon normal genes within the placenta and how this may affect fetal development. So in Southampton, we have the low protein mouse model, and we see that a low protein diet during pre-implantation leads to effects on the offspring, so fetal growth restriction leading to postnatal increased weight and increased blood pressure. But what we notice is that at the blastocyst stage, this diet leads to a reduction in the number of perfectoderm cells, a reduced placental size, and when we look at the gene expression of these placentas, there are changes to gene expression associated with changes to DNA methylation. So this is suggesting that there is this link between changes to the placental epigenetic status, its gene expression, its function, and how it programs the phenotype of the individual. So what about in humans? So in Southampton, we have the Southampton Women's Survey, where we've got highly characterized pregnancies with information on the maternal environment in terms of diet, lifestyle, and maternal blood measures. And what we can do is we can relate this to aspects of placental function and to fetal development. So what we found is by looking at one example of placental function is amino acid transport. And so what we found is that actually maternal body composition, such as BMI or maternal muscle mass, relates to the expression and the activity of amino acid transport in the placenta. And then when we look at these amino acid transporters, they in fact relate to the size and the lean mass of the individual. So what we're interested in is how are factors in the placenta being regulated by the environment? And one thing we found was that maternal vitamin D levels related strongly to the expression of various genes in the placenta that related to transport function. So we're interested in vitamin D because we know that maternal vitamin D levels are associated with changes to fetal size, but also the programming of not only bone development, but of obesity in the child and in the adult. So we wondered whether these effects are actually being mediated by the placenta. So vitamin D in its inactive and active form being taken up by the placenta, and actually having transcriptional and epigenetic effects within the placenta. So vitamin D will act by the vitamin D receptor and the RXR heterodimer to switch on genes, therefore leading to protein expression, which will affect placental function and impact upon the fetus. So that's our working hypothesis. But actually, vitamin D can have effects on gene expression in several ways. So vitamin D can activate the vitamin D receptor to form this heterodimer with RXR, which can then switch on the transcription of various genes for growth promotion and protection against the inflammatory response. But all these factors that promote the transcription via vitamin D can also be methylated. So there is this idea that these factors not only are methylated, but also the vitamin D will activate the vitamin D receptor, which will activate other proteins that will change the chromatin structure, which will lead to DNA methylation itself. So to follow up these ideas, we cultured human placenta with vitamin D, and we looked at the transcription, so the messenger RNA levels by RNA sequencing. We looked at the DNA methylation by 850K array, and we looked at the proteomics in response to this vitamin D culture. And so we found that there were large effects of vitamin D on the gene expression in the placenta. So we had both upregulated and downregulated genes in response to vitamin D. We also had changes to the methylation of the DNA in specific genes, and we had corresponding protein changes. And all of these corresponded in terms of the pathway analysis to actually lead to increased chromatin remodeling. So there was something going on that was changing the global epigenetic regulation, allowing the gene transcription to take place. And we were dampening down the inflammation response in the placenta. So this was suggesting that the vitamin D is having not only growth promoting effects, but it's also having a protective effect. So the interesting thing we found when we looked at our placenta cytotrophoblast cell culture doing a ChIP-seq experiment was that we actually found that there were no differences in terms of vitamin D to the overall chromatin structure in this short exposure. But what we found was that any responses we saw to vitamin D were dependent in the underlying epigenetic landscape. So there were tissue-specific effects to transcription that would only take place in a certain permissive state of the chromatin. And this, as I say, this was specific only to placental tissue. So what we found was that, yes, there were changes to gene expression, there were changes to DNA methylation, but there was also this underlying epigenetic background which determined how this tissue could respond to vitamin D. So in this study, we therefore highlighted targets which we could go back into our cohorts and we could see how they related to fetal development. Indeed, we found that key factors in this vitamin D handling pathway, such as the RXR and the vitamin D receptor, indeed related to the size of the fetus at birth and during childhood up to 4 years of age. So we saw it impacted upon their development of fat mass as an individual. And so we're now following this up in a study where we're actually giving women vitamin D during pregnancy and seeing how this impacts upon the development of the fetus, but also the effects on the placenta in terms of gene expression and in terms of the epigenetic profile in terms of DNA methylation. So overall, we've shown that in our specific studies that effects in the maternal environment can impact upon the gene expression profile of the placenta, and this can be via changes to the epigenetic regulation, which may be therefore leading to these long-term effects on gene expression, which can lead to changes in terms of the placental phenotype, but also effects upon the fetus and its increased risk of disease in later life. So I just wanted to acknowledge all of the people that contributed to this study, and also obviously the patients and the midwives that helped us with this sample collection in terms of the human placenta. So thank you.
Thank you. That was very, very informative and very nice. Thank you very much, Jane. There are many questions, some comments in the chat. So I would like to ask one of the questions is from Paola Valero. It says, thank you very much. When you talk about methylation of key genes during pregnancy, these are regulated by the mother's environment, but the father provides at least half of the genetic information of the baby. Is there any report on whether the father transmits any information that affects the methylation of the genes of the placenta?
Yeah, there are papers out there definitely showing in the mouse, in that same mouse model I presented, and in human studies showing that the methylation profile of the sperm will impact upon the phenotype of the individual. Whereas in my studies, I'm thinking I'm thinking of during pregnancy. So I'm looking at direct effects of the maternal environment during pregnancy upon the changes to the placenta and the fetus. So I haven't really looked myself at the paternal effects.
Good. There's another one. This is from Marcelo Cornejo Alanis. Thanks for the beautiful presentation. The effect of metabolic disorders in clinic is critical in the development of the fetus. Thinking about early prevention, what are the candidates for early diagnosis and prevention? Sorry, I lost that. Prevention. Which metabolite is possible to identify at the gene level?
That's something we're interested in. So that's why we are obviously characterizing normal pregnancy, so normal placentas. We're looking at placentas from sort of clinical situations, and we're using our ex vivo preps to change the environment. And then we're trying to look at what things are changing in the placenta with the hope that we can map these to metabolites that are being perhaps excreted back into the maternal circulation, or vesicles and microRNAs and that sort of thing. So we're not at that stage yet, but that would be our hope, to find targets that we can identify. And lots of people are looking at this and probably have already identified possible targets. Okay.
Thank you very much. There's another comment from Sandra Cortez from Chile. She says, amazing lecture, which is very nice.
Thank you. Wow.
Okay. There's another question from Kenny Araujo. He's from Venezuela, but he's now in Chile. He says, thank you, Dr. Jane. Can the deficiency of other vitamins produce these epigenetic changes?
Well, I know zinc can affect epigenetic changes, and obviously there's effects of the folate, but there's various different nutrients that can impact upon the epigenetic status within any tissue. But we were just very interested in vitamin D because we were seeing these associations with fetal development, but actually no one had really looked at how it was affecting the placenta itself, and we're just assuming that it was passing straight through and that it was having no effect at all on the gene expression or any methylation or anything. So that's why we targeted that. Obviously, we would like to look at other, we have looked at some other nutrients, and we would be wanting to look at other things as well.
Okay, and I think one of the final questions for this session is, for this section of the session today, is from Carlos Palma. Thanks for the presentation. When you talk about maternal environment, what or how long would you consider acute rather than chronic exposure of the placenta?
That's a really interesting question, and that's something we want to tease out, because in our lab preps, we're obviously just taking ex vivo samples. We're doing 8 hours. In our cell preps, we can do up to 48 hours. And in our cohorts, we're looking at vitamin E supplementation throughout the whole of pregnancy. But then we also have the impact of seasonal effects, and we have all the other things adding on to it. So actually, I think it might be something that we need to model computationally to try and work out what the levels of effects are in different time spans, with all of the other factors sort of accounted for. So that's the whole study in itself.
Good. Have you had the option of seeing patients and epigenetic changes in the placenta now that we are passing the COVID-19? Is the same phenomenon going to happen? Is something different going to happen after these patients that were infected today?
I imagine so, because I know there's a UK collection there's a UK study looking at collecting placentas and looking at the impact of COVID on those in terms of just transmission and effect on the phenotype. So I imagine they've already got that covered in terms of the methylation effects.
Good. Okay. Okay, thank you very much, Jane. That was very nice. And we will move on now with Dr. Carlos Escande. He's from the Institute Pasteur from Montevideo in Uruguay. He's a PI, he's a principal investigator at the Institute Pasteur, and he has been trained in metabolic disorders mainly. He, in fact, studied obesity and type 2 diabetes at Mayo Clinic at the United States, and is working he founded, in fact, a startup company, which is now based in Uruguay and the States, called Iolo Pharma. So we have a different view of the metabolic problems, and I think it's very interesting. So Carlos, thank you very much for accepting being here, and it's all yours, the screen, not the stage.
Thank you very much, Luis. First, I want to thank you for organizing this amazing course, with having people around the world and students all around the world coming together. I think it's really, really nice initiative. This is one of the things that we need to thank also to the pandemics, this kind of new way of communicating that I think we have now. Thank you for the introduction. What I'm gonna show you today is part of the research that we have been conducting here in the Institute Pasteur in Montevideo. As Luis was mentioning, this is also we also started as a small startup company in order to be able to fund the research that I'm going to show you. So there's a conflict of interest statement that I'm going to show you later. So, but my talk is going to be, let me see, there we go. Focus on obesity, as this is, I mean, the main concern that we have been focusing in the past, I would say 10, 15 years since I started my PhD studies in the US. As you all know, obesity is a huge problem all around the world. I mean, the numbers of obesity and how it affects the human health are amazing. I mean, you can see here that this is how obesity has been increasing over the years since the 1970s, and it has been growing non-stop. This traditionally has been seen as a problem from developed countries like US, some European countries, but this is something that is affecting all, I mean, except from some very poor countries, I would say the whole world. In Uruguay, we have numbers that are very similar to the Western countries. We have like 65% of adults and 40% of our children are either overweight or obese. So we are facing a huge problem now and also for the future. And as you probably all know, obesity is the main the main driving force for type 2 diabetes. The numbers of type 2 diabetes have duplicated since the 80s. It's calculated that around 400 million people are currently living with type 2 diabetes. And the healthcare cost for the treatment of obesity, diabetes, and all the related metabolic disorders are huge. So this is a big, not only health problem, but also economic problem for most countries. We have been, I mean, working with trying to understand obesity and inflammation with a main focus on adipose tissue. We have been developing a lot of different lines of research, and just mentioning some of the things that we have been doing in the past 10 years. We have been working with this protein DBC1 and how regulates obesity and tissue inflammation. Also with the protein C38, that is one of the main NAD glycohydrolases, and how NAD therapy is emerging as a possible way to treat metabolic disorders and also aging disorders. And more recently, we have been starting to do some research on how, 36 regulates inflammation during obesity. But this is all published. I'm not going to focus on this. I'm going to focus on some work that we have been developed, trying to develop new drugs for the treatment of obesity and type 2 diabetes. And as I was mentioning before, I have to declare a conflict of interest that we started this small company in Pasteur that is financing the research that I'm going to show you. So one of the main landmarks of the transition from obesity to the metabolic disorders is the establishment of a chronic environment of inflammation. And this is low-grade inflammation. It's very different from the acute inflammatory response that we face when we get an infection. This is a state that it does not resolve by itself, and it has been shown that plays a key role in the development of type 2 diabetes, atherosclerosis, and so on during obesity. And one of the key factors of this inflammatory response is the activation of the transcription factor NF-kappa B. This is a very simplified cartoon that I'm showing, but it is a very schematic view how these things work, right? So we have obesity. This leads to activation of NF-kappa B, and here we are putting the activation of NLRP3 inflammasome, but there are also other inflammatory pathways that are being upregulated during chronic inflammation in obesity, and this leads to secretion of cytokines, and leading to a positive feedback with other tissues that eventually will contribute to the development of type 2 diabetes and other metabolic disorders. And some years ago, the group that is being led by Bruce Freeman, originally in Birmingham, Alabama, and now in Pittsburgh, discovered these molecules that are called nitro fatty acids. That is basically a fatty acid bound to a nitroalkene group that reacts especially with cysteines and histidines in proteins from a Michael addition reaction. And it has been shown that this active nitroalkene molecule has very strong protective signaling pathway, activates a signaling pathway, sorry. So here is a scheme of what nitroalkenes do. These nitrophatty acids do. They inhibit NF-kappa B dependent transcription. They activate heat shock response. They activate NRF2, and they also regulate PPAR gamma signaling. And something that is very interesting about these molecules is that they have been tested in a lot of animal models of different metabolic diseases. And here is just a scheme of what you can see the effects in atherosclerosis, hypertension, type 2 diabetes, and so on. So they were very promising molecules, and actually some of these molecules have been taken into clinical trials, at least they have gone through phase 1 and phase 2 clinical trials for some metabolic diseases. But these molecules, and coming back to this, the most important part is the capacity of this nitroalkene group to react with proteins and activate different signaling pathways. They have some problems, especially they are very rapidly excreted and metabolized by the mitochondria. They are very rapidly saturated. So there were some issues with these molecules that we thought that they could be improved. So this is where we came with this. It was at the beginning a very simplistic idea, and I'm going to show you that it has become more and more complicated. But the idea that we had when we started this project was, okay, let's take this very small nitroalkene group that we know that has very strong anti-inflammatory properties, and put it in different in novel scaffolds, right? So, and the idea behind this it was, okay, let's try to get the activation conferred by the nitroalkene group, but try to use different scaffolds to improve their actions. And of course, the idea was always try to approach metabolic syndrome, insulin resistance, obesity, and so on. For that, we use this is the mouse model that we use. It's a standard mouse model for diet-induced obesity and preclinical evaluation of drugs. Basically, it's C57 mice that are fed with a high-fat diet. This is the composition of the diet, where basically what we do is to increase the amount of fat that we gave, and we decrease the amount of protein and the carbohydrates. So the amount of calories that we give to the mice is the same. The only difference is the composition of the diet that has more fat, and the mice become obese because of that. And is the most accepted model for obesity, liver steatosis, or liver steatosis, glucose intolerance, and insulin resistance. It has, as all animal models, its complications, sometimes translation and many times I would say from the mice to the humans is not very simple, but it's still the most accepted model, animal model to test drugs in obesity. So, and this is the first molecule that we developed. This is what you are seeing here is vitamin E, alpha tocopherol, that we added this small nitrogen group to the molecule. And the idea here was to see if we could use this molecule to treat atherosclerosis, because vitamin E incorporates into the lipoproteins, and it was supposed to go to the lesions in atherosclerosis mice models. And this is just a summary of what we found, is that by adding this small molecule to the alpha tocopherol, we were able to decrease inflammation in the site of the lesion, and we were able to decrease the amount of lesions. This is what I'm showing you here is the aorta stained for lipids, and you can see here how a normal mouse developed a lot of plaques here, and the amount of plaques that you see when we treat the mice with our molecule. And here we were treating the mice with alpha tocopherol, right? So the only difference is this small nitrogen group that we added to the molecule. Next, in the next step, we wanted to try to get a more potent molecule based on the alpha tocopherol. So what we did was to remove the isoprenyl tail from the vitamin E and add this the nitrogen group to the short tail that we left behind. And what we were trying to get here was a molecule that was more hydrosoluble and would go to all tissues and have a better biodistribution, which in fact we were able to get. This drug goes beyond the lipoproteins. It goes to the liver, to the muscle, to the adipose tissue, and so on. And we were able to show that, again, in a model of diet-induced obesity, we decreased inflammation, although there were no changes in body weight of the mice when we treated with the drug. There was a decrease in inflammation. There was some improvement in glucose tolerance. So we were able to show that by giving this molecule to mice that were already obese, we were able to inhibit NF-kappa B and somehow make us dissociate the obesity from the this pro-inflammatory state of the mouse, and by doing so, the decrease glucose intolerance and reduce the amount of type 2 diabetes. So this is I'm showing you this very fast as what I want to show is the proof of concept that we were trying to develop. That we could take this very, very small reactive molecule, nitrogen group, put it into different molecular scaffolds, and see if we could use this strategy to make better and better and better and better drugs to treat type 2 diabetes and inflammation. So coming back to this, so we had these two molecules that we knew that they were working, but we wanted to keep improving our drugs. So we decided to take a step further, and we completely walked away from vitamin E. And so when we were trying to select a novel scaffold to add the nitrogen group, we decided to take these three things into consideration. First, to know that the drug was safe in humans, right? That it had been already tested in humans, yes, and it was known that was safe. And the other thing that we asked the scaffold was to that it was known that there had positive metabolic actions, that humans had used the drugs, and there was proof that there was an improvement in metabolic actions. And of course, we were asking the scaffold, if possible, to be an NF-kappa B inhibitor by itself. The idea was to, okay, let's take a molecule that has been known, that has been used in humans, that has anti-inflammatory actions, and try to improve this drug by adding this, let's say, to potentiate these actions by adding the nitrogen group to this molecule. And this is how we came to this, one of the, I would say, most ancient drugs that has been used by humanity, that is salicylate. The salicylate is a precursor of aspirin. It has been used to treat inflammatory conditions for centuries. And actually, there was a rebirth of salicylate a few years ago. In 2004, it was shown that salicylate inhibits NF-kappa B by itself. Later, it was shown in 2012 that salicylate activates AMPK, that is a master regulator of energy metabolism, is one of the key kinases that stimulates catabolism in mouse and humans. And actually, there were several clinical trials using salicylate in patients with type 2 diabetes, and they showed somehow beneficiary effects on the patients. The main concern about salicylate was that they had to be taken in very high doses, but it was actually able to reduce free fatty acids, to reduce triglycerides, to reduce basal glycemia in patients with type 2 diabetes. And actually, it was taken for up to 36 months, and there were no important side effects of the drugs. And the last clinical trial was from 5 years ago. So this is something that is not old science, right? There was a rebirth of salicylate. So we decided to add this nitrogen group to salicylate, and we generated this molecule that we call SANA. And I have to say, when we first started testing the molecule in vitro, we saw that it had some improvement in the anti-inflammatory action. It was able to inhibit NF-kappa B. It was able to inhibit TNF-alpha secretion, IL-1 beta secretion, but it was not great. I mean, the doses that we needed to get the drug to work as an anti-inflammatory molecule were kind of high also. So we were not very excited about the molecule at the beginning. But when we put the mice on a high-fat diet and we started giving the molecule to the mice, we found this one was an amazing effect from my experience in terms of weight gain. So we here what you can see in green is the mice fed on a high-fat diet and with salicylate, different doses of salicylate from 200 to 400 milligrams per kilo per day. And when we started giving our drug, also there was a clear dose dependence in prevention against obesity. And when we got to the maximal dose, there was a complete prevention against weight gain. So the mice were exactly the same as the lean mice, and there were no side effects whatsoever. So it was a complete protection against diet-induced obesity when we gave the drug. And I'm not going to show you the data, but we were able to low down the dose to 10 milligrams per kilo per day by changing the solubility of the drug and improving some the formulation. So we are talking about the drug that is working at around 40 times more potent than the scaffold that is the salicylate. We found that the protection against obesity is, of course, because a decrease in fat accumulation. You can see a mouse here fed with high-fat diet. We are showing the fat depots here. We fed with salicylate. There is no difference between control and salicylate. And when we feed the mice with our drug, there is the fat deposition is the normal for a lean mouse, and we have tested this in all different fat depots, and there is a clear difference in fat accumulation. And the most important part is that when we test for glucose tolerance, liver steatosis, insulin resistance, and so on, we have found the complete protection against every single aspect of metabolic disorder. So when we measure glucose, is the same as a lean mouse, the glucose tolerance is the same. We have decreased insulin levels, free fatty acids. There is a complete protection against liver steatosis. So it's like the mouse was being fed on a normal chow. It seems that they don't see the high-fat diet, right? This is a very, very strong effect that we see with the drug. And not only that, there is always the critique about how to translate the drugs from mice to humans, because you don't give the drug to a lean individual. You are giving you want to treat a human that is already obese. So we make the mice obese, and then we started giving the drug, and we see the same. So with this, we see that in obese mice, we can see a decrease in weight gain. You can see here the pictures. The mice that were already obese when we started treating, they lose weight, they improve their glucose, they improve their liver steatosis, they improve their markers of liver damage. Not only that, we compared the drug with metformin, that is the gold standard for the treatment of type 2 diabetes. And I'm going to focus here in how the evolution of glucose level. The mice that were on a high-fat diet, they increased their glycemia over time. Salicylate and metformin, they have an effect of stopping the increase in glucose levels in the mice. But our drug, and in combination with metformin, was able to decrease glucose levels in obese mice. So we think that our drug is clearly more potent than metformin, that is the gold standard for the treatment of type 2 diabetes in humans. So the question that we wanted to ask is, okay, so how are we preventing obese, or how are we making the mice to lose weight? And obesity, it comes from this balance from the calorie intake compared with energy expenditure. The only way to become obese is that when the amount of calories that we take is higher than the total amount of energy that we waste, right? And how can we achieve changes in that? Either by decreasing food intake, decreasing absorption of nutrients, or by increasing energy expenditure. That it means increasing the amount of activity, like let's say go out and running, or increasing the resting energy expenditure. This is what we thought it was happening, because we checked food intake, absorption, and we checked activity on the mice, and we saw no difference at all. So we put the mice on a closed system that we can measure oxygen consumption, and we saw that the mice treated with the drug, they have an increase in oxygen consumption, meaning that they have an increased metabolic rate. We wanted to know where this increase in metabolic rate was happening, and we saw that the main tissue that had an increase in metabolic rate, and this is measured by uptake of FDG as an analog of glucose, was in sub-Q adipose tissue. And this brought us to, and I'm going to go fast here, I'm about to get out of time, to thermogenesis. And thermogenesis is a physiological mechanism in all mammals to maintain body temperature. Humans have brown adipose tissue, and there is a lot of research being developed trying to induce white adipose tissue, that is the main tissue that we accumulate in obesity, into somehow a brown tissue. This is called beige or brown, or yes, it's called beige. That what we make here when we induce browning of adipose tissue, of white adipose tissue, is to make this white tissue converted from a tissue specialized in storing energy into producing heat and reducing the amount of energy that is being stored. I'm gonna go fast here. We found that actually this is the way that we test if there is increased thermogenic capacity in mice. We have mice at 22 degrees, and we put it at 4 degrees in the cold and see how the temperature drops during that time. And we saw that the mice treated with our drug were capable of maintaining body temperature, meaning that they had increased thermogenic capacity. And I'm gonna briefly comment on this. There are many different mechanisms that induce thermogenesis. Most of them involve the mitochondria. This is the classic UCP1 dependent thermogenic pathway that uses where the mitochondria uses the electron transport, the electrons transported through the inner mitochondrial membrane to produce heat. And this is a novel mechanism that has been described a few years ago, that uses creatine as a futile cycle of phosphorylation, a dephosphorylation of creatine, and this cycle produces heat. We used quantitative proteomics to try to pinpoint which was the mechanism that was being induced by our drug. This is proteomic data on adipose tissue and proteomic data on isolated mitochondria from white adipose tissue. And we found that this is the pathway that is being stimulated by our drug, that is creatine-dependent thermogenesis. And this is the first time that there is a drug that activates this pathway. So I think we have our first-in-class drug to stimulating this thermogenic pathway. And actually, we showed that if we use a creatine inhibitor, we abolish the effect of our drug on cold tolerance. So sorry, this is the pathway that we are proposing that is induced by our drug. And finally, just to let you know, we have just finished all the FDA requirements to approve for a phase 1B clinical trial. So we are on the go to start giving the drug to humans. The idea is to give starting the second semester of this year to start testing the drugs in humans. So this is just the acknowledgements, my lab, and Iolo Pharma, that is the company that is financing this last part of the research. Thank you.
Okay, thank you very much, Carlos. Interesting and very impressive how you move into basic science and then application. Thank you very much for that. There are many questions, not much time left, but I will go very fast, and if you can give also short answers. For example, Hector Ramos is asking what are the possible serious adverse effects that could result from the use of SANA? Adverse effects of SANA? Hello? I think he's frozen now. You may come back. All right, so what we can do is just leave the question here, and he will answer the question when he rejoin the session now, unfortunately. One more go, Dr. Escande? No. Okay, I think he left. Right, so we will move then to, here he's back. Are you there now?
Sorry, I lost my connection. I really apologize.
I was just saying that very quick answers to the questions because we have no much time now. And Hector Ramos was asking adverse effects of SANA. What do you think about?
This is we are working a lot around that because we need to know which there are always adverse effects of drugs. We all know that. We know that at high doses it can induce some kidney damage. We are talking about 10 times the effective dose. This kidney damage is reversible if you stop administration, and we also know that it can induce some stomach damage if taken in high doses. This is something that can be avoided by changing formulation. So this is where we are now.
But
this is what we know about the side effect of the drugs.
Fine. Carolina Guevara is asking, how did SANA impact insulin levels and insulin resistance in the mice model?
Yes, the insulin levels went down to normal, and insulin resistance was in our mice models, it was completely corrected even in the mice that were already obese when we started treating the drug. This is why we are so excited about the drug.
Right, good. Professor Van Hoor from the Netherlands is great work. What is the concentration of SANA used in these mice, and can these concentrations be used in humans? That's the question.
This is a very good question. So we were able to, salicylate was given to patients in 1 to 5 grams per day. This is the same kind of dose that you have to give in mice. We were able to decrease we were able to go as low as 10 milligrams per kilo per day to the mice, which in translation to humans would mean around 500 milligrams, a pill of 500 milligrams for humans.
Good.
So
There's
this
so many
is
questions
a dose that can be given to a human being. Yeah.
All right. There's so many questions and good questions. I don't want to miss anyone, but I don't have more time. Anna Elias is asking, these molecules can improve inflammation and metabolic parameters, but not the weight. This could result in a metabolic healthy obese phenotype. What do you think?
we believe that all the metabolic improvement that we get comes from the effect on obesity. Actually, when we developed the drug, we were going after that, and let's say a healthy obese individual with no inflammation. But we see this weight loss, and we think that most of the effects are because of the decrease in the adipose tissue content.
Right.
Yep.
Dalmiro Gomez asking whether you think that PPAR receptors are involved in the mechanism of action of these anaboles.
We are working as we speak, trying to find the molecular targets of the drug. This is something we still don't know. We have some clues, but we still don't know. But we tested all the known targets for nitrofatty acids, and we know that PPAR gamma is not a target for our drug.
And the final question I'm asking is, because she was the first asking, Sofia Villalta from the Netherlands, do you think we could expect an effect in prevention of metabolic disorders considering a healthy obesity condition or overweight?
Can you say the question again? I didn't quite get it.
The question is whether do you think that we could expect an effect in prevention of metabolic disorders considering a healthy obesity condition or overweight, for example?
I hope so. I mean, achieving weight loss in humans is really hard. I mean, the only drugs that have good effects in weight loss are the GLP-1 agonists so far that are in the clinic. We are confident that we may be able to get an effect in humans in weight loss, but even if we don't, we would be really happy if we can get into a, let's say, healthy obese individual. This is not what we are aiming. We are aiming for obesity, but still, I mean, it could be that is not strong to induce weight loss, but still helping in preventing inflammation and so on. But we still don't know.
Okay, thank you very much for your answer, your presentation.
No, thank you all.
Thank you. And I now have the pleasure of introducing to you Professor Leslie Maya. We all know Professor Maya for many years, working in the placenta and fetal and development, etc. He is Professor of Obstetrics and Gynecology at the Oregon Health and Science University at the moment. He has been also at Cincinnati and working at the University of Texas Health Science Center in San Antonio. And he's been editor of Placenta and other journals, and also president of the Perinatal Research Society, IFPA, which is International Federation of Placenta Association, and president also of the Society for Gynecologic Investigation, SGI. So thank you very much, Les, for your time, and you have 10 minutes to talk. No, sorry, you have.
Thank you, Luis. Can you see my screen, Luis? Can you see my screen, Luis?
Everything fine.
Okay, thank you. So first of all, thank you for the.
Do the full projection of the screen using that one.
You got it?
Yeah, good. We got two now.
You got two?
Okay.
Can hang on a second. That better?
That's better. That's fine. Thank you.
Okay, thank you. The pleasures of working with two screens. So first of all, let me thank you for the very kind invitation to participate in this session. And I'm going to talk about the effect and consequence of maternal obesity and gestational diabetes that's been introduced to you today. And I'm going to look at the effect on placental energy metabolism and function. And particularly, I'm going to emphasize throughout this talk the sexual dimorphism in response. And it does matter whether the placenta is male or female. So as Dr. Cleal introduced, this is the placenta, which serves as the immune barrier, obviously, between the mother and the fetus, also serves for exchange of oxygen and carbon dioxide between mother and fetus. But one of the most important functions of the placenta is that it produces prodigious amounts of peptide and steroid hormones each day, gram amounts, particularly of peptide hormones. These released to the maternal circulation where they affect maternal metabolism to actually increase the availability of glucose, fatty acids, amino acids, which can be then transported across to the placenta to the fetus. And this is obviously also influenced by the plane of nutrition that the pregnant woman is experiencing, whether she's in good nutrition, whether she's starving, whether we've got the settings of obesity or diabetes and gestational diabetes. These substrates are taken up by the placenta by a series of specific transporters on the syncytial trophoblast surface. They can either be transported straight across to the fetus, they can be stored in the placenta and buffered for subsequent movement, or they can be metabolized in the placenta. And one of the things, obviously in metabolism, they generate the ATP, which drives the peptide and steroid hormone production, but also supplies the energy to power a lot of the transporters on the syncytial trophoblast surface. But the net result of the transport and the action of peptide and steroid hormones is to control fetal growth and development, and as you've heard before, fetal programming. So I want to emphasize the placenta is not just a conduit. The placenta is second only to the brain in the number of gene transcripts it expresses. So to me, that really emphasizes its importance in growth and development. And it consumes about 50% of the oxygen and 30 to 50% of the glucose that's supplied to the uterus. Its metabolic activity is 4 to 6-fold higher per unit weight than that of the fetus itself. So at term, a placenta weighs about 600 grams, the fetus weighs about 3.6 kilos. So you can see the metabolic activity is roughly split, or the metabolic activity roughly split between the placenta, the much smaller placenta, and the fetus. And in the placenta, a third of the placental oxygen consumed is used for de novo generation of peptides, 1/3 to maintain that cation gradient across the membrane necessary for transport, and 1/3 for other uses. So again, to emphasize, it's not simply a conduit, but it regulates nutrient composition and supply from the mother to the fetus. And I raise this question here, is it a selfish organ? So by virtue of the amount of metabolites or nutrients that the placenta and oxygen the placenta consumes itself, does that alter what's then available to the fetus? So David Barker and Erickson coined this phrase about 15 years ago, that boys live dangerously in the womb. And that's because we know there are different evolutionary strategies for males and females, that the male fetus wants to keep growing in utero, and they're usually larger at term, but males have many more adverse outcomes due to then less placental adaptability. So in their desire to grow larger, it means they've often got less placental reserve capacity, less adaptability when faced with stressors. And that means they've got more adverse outcomes, such as preterm birth, preterm rupture of the membranes, placenta previas, preeclampsia. Their lungs don't develop as well. They're often more macrosomic or large male fetuses. There's a lot more late stillbirths, and they actually, the effect of a male placenta is that there's poorer maternal beta cell function and an increased risk of gestational diabetes. But females have a more reserved growth strategy. They adapt their growth rate to optimize survival, particularly in a poor environment. So they're obviously determined for propagation of the species. They have reduced growth trajectory. That means they have increased placental reserve capacity, so that when some insult or stressor comes along, they've got greater adaptability to be able to survive that stress. And these effects are also reflected by differences in fetal programming of males versus females. There's a lot of evidence now for sexual dimorphism in placental function. There are quite a few papers now showing differences in gene expression, both in the first trimester and at term. And these have been linked to an escape from X chromosome inactivation. But we and many, many other groups have shown sexually dimorphic differences, say in inflammatory, hypoxic, apoptotic, or autophagy responses, in the expression of antioxidant defense enzymes, fatty acid transporters, and fatty acid oxidation, the response to adiposity and inflammation, differences in microRNAs, and steroid and peptide hormone synthesis, and all of these have been linked to the differences in outcomes seen in male versus female fetuses. So I'm going to talk more about the maternal metabolic milieu of obesity and gestational diabetes. We know we see insulin resistance, hypoglycemia, and hyperlipidemia. We see inflammation, oxidative stress. Again, these responses are sexually dimorphic. And again, obesity and gestational diabetes associated with adverse outcomes, including stillbirth that I'll come back to. And again, both conditions program the offspring for disease in later life. And what I'm going to explore a little bit is there in the face of hypoglycemia, hyperlipidemia, is there metabolic reprogramming in the placenta, i.e. are there changes in cellular bioenergetics to adapt to these changing environmental conditions? And a classic example of this might be cancer, where we see the so-called Warburg effect, where we get aerobic glycolysis and altered mitochondrial metabolism in tumor cells. So we studied this mainly using the seahorse instrument, using the so-called mito stress kit. And the seahorse instrument, in fact, is quite a fancy plate reader, and it essentially can measure dissolved oxygen content in cell culture medium and pH. And we can actually have cells in a culture plate, and we can measure their oxygen consumption rate. So here we have cells sitting quite happily. We can measure their basal respiration or their basal oxygen consumption rate. If we then inhibit complex 5 or ATP synthase in the electron transport chain, oxygen consumption falls. And this is the amount of oxygen coupled to ATP generation. If we then totally uncouple the electron transport chain with FCCP, we get what's called maximal respiration. And the difference between basal and maximal is so-called spare respiratory capacity. So if I or you are being chased by a tiger, we want to have good spare respiratory capacity. That's our ability to respond to stress, okay, our mitochondrial response to stress. If we then kill our mitochondrial electron transport chain totally by adding antimycin A or rotenone, what we're left with then is solely non-mitochondrial respiration in cells. So we can apply this mito stress test to trophoblast cells from placentas from various conditions. And what we originally did was we actually looked at the effect of increasing maternal BMI on trophoblast mitochondrial respiration. And you see here that if you look at maternal BMI from 18.5 to 45, we see a adiposity-dependent decrease in basal respiration, maximal respiration, spare capacity, ATP-coupled respiration, non-mitochondrial respiration. And this is about the order of about 50% reduction in basal respiration between a maternal BMI of 18.5 to 45. What happens then if you add gestational diabetes on top of this? And we know obviously that obesity predisposes women towards gestational diabetes. So here we're comparing women with type A2 gestational diabetes, which is severe gestational diabetes that has to be controlled by administration of insulin, as opposed to A1 gestational diabetes, which is controlled just by diet alone. And this is compared to control. And in this setting, control are actually women who have a BMI of around 32. So they're actually obese. But you see with the addition of A2-GDM, so these are obese plus A2-GDM, a further significant decrease in basal and ATP-coupled respiration and maximal respiration. But there is no additional effect of the milder form of gestational diabetes, A1, versus the obese alone. But these cells then show metabolic flexibility, and they're able to increase glycolysis. So here we're measuring expression of GLUT1, the major glucose transporter on the surface of syncytial trophoblast, and then hexokinase 2, phosphofructokinase, and LDH enzymes involved in glycolysis. And you see that as we go from, again, control obese to A1 gestational diabetes to A2, that with A2 gestational diabetes, significant increase in glycolysis and no significant effect of A1 gestational diabetes. But saying in the face of severe gestational diabetes, we see metabolic flexibility, metabolic change in these cells, so they're able to switch from mitochondrial respiration to glycolysis to try and maintain ATP production. So what's the clinical implications of these findings? Well, obviously, as I've said, mitochondrial respiration is compromised by increasing adiposity and A2 gestational diabetes. But interestingly, in our studies, all these pregnancies went to term and they had good outcomes. However, there may be a cost in later life for offspring and mother by fetal programming and by the effect of the obesity and gestational diabetes on the maternal cardiovascular system. And so you might ask, though, but does compromised mitochondrial respiration matter if all these pregnancies went to term with good outcomes? So is there sufficient placental reserve, and actually could they increase their glycolysis enough to actually compensate for the reduced mitochondrial respiration? But what might happen if there's an additional insult or you actually exceed the reserve? And we pose the question of, could this be associated with stillbirth? And shown here is data from studies done in the United States, where it's the risk of stillbirth by gestational period and the effect of BMI, so increase in adiposity. And here we have the hazard ratio for stillbirth compared to women of normal weight. And you see here we're in late gestation, and with increase in maternal adiposity going here from overweight through to class 1, class 2, class 3 obesity, and then a BMI greater than 50, we see a significant increase and a quite profound increase in late stillbirths with increase in adiposity. So it goes up to about 9-fold greater in late gestation with a BMI greater than 50. So it's possible that this is associated with this decreased mitochondrial respiration we're seeing. So we then wanted to examine the use of different nutrients by the placenta and how they supported ATP generation. So the dogma has always been that glucose is the major substrate used by the placenta and fetus to maintain growth, but they can obviously use fatty acids and amino acids as well. Each of these is taken up by specific transporters on the trophoblast surface and can be transferred across the trophoblast and effluxed into the fetal circulation, but they can also be stored or buffered for later usage, or they can also participate in the generation of ATP by the electron transport chain. And this is due to their metabolism into the TCA cycle, and then they provide the reducing equivalent to drive electron transport chain. So how can we look at the contribution of each of these different nutrients to overall ATP generation? So we take advantage of the use of specific inhibitors of each of these pathways. So we can add BPTES, which is an inhibitor of glutaminase, UK5099, which inhibits the mitochondrial pyruvate carrier here, or etoxamia, which actually inhibits CPT1 alpha, the carnitine palmitotransferase, which transfers CPT1 across the mitochondrial membrane to make it a substrate for beta oxidation. So if we use these inhibitors in isolation or combination, we can dissect the contribution of each of these nutrients to overall ATP generation. And that's so-called the mitochondrial fuel flex assay, and we can measure fuel dependency. So that's the measurement of the cell's reliance on a particular fuel pathway to maintain baseline respiration, so rest in respiration. We can also measure capacity, the ability of a cell to switch and oxidize another fuel when other fuel pathways are inhibited. So if we inhibit two pathways, can it move to the third pathway and support ATP generation? And the difference between capacity and dependency is what we call flexibility, the ability of a cell to increase oxidation of a particular fuel to compensate for inhibition of the alternate pathways. So what we saw here, and we published this paper in 2019, we've got in the green, capacity, blue is dependency, orange is flexibility for use of the three substrates, glucose, glutamine, or fatty acids. And the three bars we're comparing, I'm sorry, it's a little hard to see, the effect in trophoblasts from lean women, trophoblasts from obese women, and trophoblasts from women with A2 gestational diabetes. So if you just look at dependency, first of all, so as we go from lean to obese to A2 GDM, there's no difference in dependency for glucose and for fatty acids in the female trophoblast, but dependency for glutamine goes down. In comparison, when you look at male trophoblasts, no significant difference in dependency for glutamine, but as you feed, as you're giving these cells more glucose or fatty acids with obesity and gestational diabetes, they increase their dependency on them for baseline respiration. So they've adapted to use of these more available substrates. If we look at capacity here is in green, and you see differences again between male and female, but the payoff is when we come to flexibility. So as you if you look at flexibility for glucose, glutamine, or fatty acids in the female placenta, there is no difference or no effect of moving from lean to obese to A2-GDM. But what you see in the male placenta is a significant decrease in flexibility for use of either of the three substrates as you go from lean to obese to A2-GDM. So the male trophoblasts are significantly affected in terms of their flexibility. They can't move as easily between use of the three different fuels as you go from obesity to gestational diabetes. So to summarize, no difference in dependency for the three fuels in lean women between male and female. But when you get the hypoglycemia, hyperlipidemia of obesity and A2-GDM, you get increased dependency on glucose and fatty acids for baseline respiration, but only in the male placenta. So the male likes the hypoglycemia, hyperlipidemia. It uses those increased nutrients, but that's accompanied by significantly decreased flexibility then to switch between the three. And does that contribute to the increased risk of adverse outcomes? And it changes incrementally from lean to obese and to BMI matched A2-GDM, suggesting to me the effect is not due to obesity alone, but to maybe reflect the continuum of worsening hypoglycemia, hyperlipidemia from obesity to A2-GDM. But the male definitely fares more badly. I want to give a couple more examples of sexual dimorphism in placental function, particularly in the role of fatty acids in the placenta. So fatty acids are essential for brain growth, and we know that DHA and arachidonic acid are essential brain-specific fatty acids important for CNS development. Brain growth increases dramatically in the third trimester and postpartum, with significant increases in DHA and arachidonic acid, in the brain. So these are being taken up rapidly and transferred across the placenta to support the growing brain. And I want to draw your attention to this study done in 2018, where a group in London studied the effect of brain-specific fatty acid supplementation during pregnancy on brain size, and they determined that by MRI after the baby was born. And they did a double-blind placebo study and found that only the males born to the BSA-FA supplemented group had significantly larger brain volume. And it was total gray matter, corpus callosum, and cortical volumes when compared to the placebo group. So there was no effect of supplementing with these brain-specific fatty acids on the brain size of the female fetus. It only happened in the male fetus. So it seems the male is taking in more of the brain-specific fatty acids. So we looked at, did a lipidomic analysis looking at the effect of obesity and gestational diabetes versus lean on lipid composition of the placenta. And we quantified 436 lipids and obviously prepared heat maps, etc., and did various comparisons. And I just want to draw your attention to these couple of slides. So here we're looking at phosphatidyl inositol species and the various fatty acid compositions. And I want to draw your attention to these down here at the bottom, which are PI species containing docosahexaenoic or DHA fatty acid chains C22-6. And what you see here is in this group, a significant decrease in the levels of the phosphatidyl inositol containing the C22-6, and it's significantly lower in the GDMs versus the lean. So, but this is only in male placental villus tissue. Similarly, if you look at triglyceride species in male villus tissue, again, significant decrease in triglyceride species containing docosahexaenoic acid. Again, it's this group here where you see this significant decrease in triglycerides that contain C22-6, particularly when you compare GDM versus lean, but also obese versus lean in the male placentas. So again, this effect, there's less of these long-chain C22-6 fatty acids in lipid species in the placenta of the male fetus compared to the female fetus. So correspondingly, if they've got less of the long chain, they're going to have more of the medium chain fatty acids relative to the female. So this just summarizes what we found here. If we look at the lipidomics, in the male placenta, we see more of the medium chain relative to the long chain, which I just told you were lower. Obviously, in the female placenta, we've got more long chain and long chain fufus compared to the medium chain. That corresponds to some work that we did as well, where we measured fatty acid enzymes involved in fatty acid beta oxidation. And we found more of the enzymes that had specificity for medium chain fatty acids in the male placenta versus the female placenta, where we saw more of the fatty acid beta oxidation enzymes that had preference for the long chain fatty acids. And that also parallels levels of fatty acids found in amniotic fluid in a study done by Catherine O'Neill in 2018, where in the amniotic fluid of the male fetus, there were more medium chain fatty acids, whereas in the amniotic fluid of the female fetus, more long chain polyunsaturated fatty acids. So definitely sexual dimorphism in lipid metabolism and lipid levels, fatty acid levels in male versus female placenta. I just want to also show you some proteomic analysis we did as well in a similar manner. So we did proteomic analysis. We recognized 2,980 proteins. We found significant differences in expression comparing the groups, and then we did gene ontology pathways to look at differentially expressed pathways using these proteins. If we look, first of all, combining the male and female placentas together, this is what you see when you compare obese to lean, A2-GDM to lean, and A2-GDM to obese. You see a significant number of pathways which are upregulated and a significant number that are downregulated here. But when you split them out to male or female alone, you see very different pathway patterns of expression. So first of all, if we look at the female placentas alone, you see pathways that are upregulated here related to extracellular region, extracellular space, cellular metabolic processes, etc. And then a series of pathways that are downregulated to do with protein transporters, centrioles, and glucose metabolic processes. Perhaps not understandably, we see these when we go to A2-GDM versus lean or A2-GDM versus obese. But in contrast, you see many, many more pathways that are affected in the male placenta, and they're very different. So there's a whole host of pathways upregulated, which are related to inflammatory response, complement activation, RAGE receptor binding, acute phase response, etc. And then a whole host of pathways that are downregulated, which are regulated to ribosomal and nuclear RNA expression and RNA processing, etc. So just to summarize, profound sexual dimorphism in the response to obesity and GDM. And do these represent sex-specific responses to inflammation, hypoglycemia, hyperlipidemia? But don't forget, the responses to these conditions are built upon the pre-existing differences in gene expression related to fetal growth and survival strategies. So in the female placenta, as I showed you, there are relatively less pathways affected, and it appears those might be driven by worsening hypoglycemia and hyperlipidemia, particularly these glucose metabolic pathways. In the male placenta, more pathways are affected, a lot to do with inflammatory responses, upregulated but downregulated, related to translation, RNA processing, splicing, nucleosomes, etc. And are these driven more by the inflammatory milieu rather than the hypoglycemia and hyperlipidemia? And again, are these adaptive changes established to maintain or enhance growth, but coming with them as a risk of demise in males versus an adaptation to ensure survival in females? So in summary, the placenta obviously has a major role in fetal programming, as you've heard. The maternal environment, particularly I've focused on obesity and diabetes, affects placental function and in a sexually dimorphic manner. And is this an adaptive response to this maternal environment? But there are many events that are altered in the placenta, and the net result is that these then impinge, again, in a sexually dimorphic manner to affect fetal growth and development. And as Jane pointed out, there are obviously epigenetic mechanisms at play in many aspects of fetal growth and development that give you the programming effect. So I want to end by acknowledging my co-workers who've helped this work over the years, and also just show you the view out of my laboratory window. If you get here early enough in the morning to watch the sunrise, this is the sunrise rising over Mount Hood. So thank you.
Okay, thank you very much. Nice and very much on time also. Thank you. We have some questions now from the people. There's a lot of questions coming in. Alvaro Santana, based on your results, should be clinical treated pregnancy disease. Sorry, the screen. Pregnancy diseases in different way depend on the gender of the baby, and also, for example, could be a preeclampsia from a male placenta different than a preeclampsia from a female placenta. Thank you.
I think there's a general consensus that that's the direction we're moving in, Luis, that you have to consider the sex of the fetus, and there may ultimately be sex-specific therapies. And again, the thing I'd like to say is we often think of placental dysfunction. Maybe we should also start thinking about placental adaptation. So if you treat something that you think is a dysfunction, but it's an adaptation, you actually might be making things worse rather than better. So I think that's something to bear in mind in future, certainly.
Yeah. Okay, there is another one from Bruna Lima, and she's saying, thank you very much. Considering the epigenetic process mentioned before, is there any particular epigenetic mechanism that could have a greater impact on this sexual dimorphism response?
Well, I'm not sure. There are obviously differences in placental gene methylation being shown with obesity. We did that quite a few years ago, and it was actually quite interesting that a lot of the changes are focused on chromosome 17 and chromosome 19, which are associated with particularly production of the placental growth hormones and chorionic somatomammotropin, etc. So there we looked at methylation, but I think again, histone acetylation is obviously probably important as well. And again, one of the things I like to emphasize is, depending on the time in pregnancy at which this insult occurs, you can get different effects as well. So a good example would be if you have pre-gestational diabetes, you enter that pregnancy already with hypoglycemia. So that might have some effect very early, an epigenetic effect very early, whereas gestational diabetes, the hypoglycemia occurs later. And so the effect then may be a completely different effect on placental function than pre-gestational diabetes.
Very true. Another one from Marcelo Cornejo. Is there any evidence of intracellular pH regulation and its association with fatty acids in patients with obesity and hypoglycemia? And
yes, yeah, I didn't have time to show all those pathways, but one of them actually was bicarbonate regulation. So again, I think pH is perhaps affected as well. It's not something we've studied particularly, but again, I think is important, certainly.
And I have one, the group that you're showing here is A2 GDM.
Yeah.
Which is treated with insulin, you mentioned.
Yes.
What about patients treated only with diet or?
Well, it looked in the studies that we did, that just the limited number of studies we did there, that the effect of diet was no different than the effect of obesity alone. These women, the women with GDM, were all matched for their level of adiposity with the obese group. And then obviously there are women who are lean women who can get gestational diabetes as well. Obviously less of them than there are obese women who get gestational diabetes. So that's an additional group we need to add in, but at least what we looked at, obese plus A1 GDM was no different than obesity alone. So the mild gestational diabetes didn't seem to have an additional effect beyond obesity. Yeah.
Good. A question from Paola Valero. One question is the male children of obese mothers or with GDM are more prone to metabolic diseases than girls?
I think that's generally correct, yes. I think if we brought Jane back, she would probably say that males are programmed more significantly, for want of a better term, and more affected than female offspring. And again, does that relate back to placental function and how does placental function relate to programming overall? So, yeah.
Good. Okay. If there is any other question, people can raise their hand. I don't know. Harry Van Hoy is on the screen now. Also, is a co-organizer of this course. Harry, you want to ask something?
No, I have no further questions. Great session today.
Excellent. Yes. Thank you very much. So. Thank you again to Les, Jane, and Carlos for the excellent lectures. And okay, we are late on time, 11:36, but we have very nice questions and answers and discussions. So thank you again, Les, Jane, and Carlos, and all the people attending today the meeting. So we meet now in the next session, which is session 4, on Tuesday, 19 at the same time, in the same channel. Thank you very much again. And please write to the lecturers. I put on the chat all the 3 emails, so please feel free to write to them directly. And please get in touch, get in contact with them and try to produce something. It's your opportunity, this course. Thank you very much. Thank you, Les. Thank you.
Bye-bye.
Thank you, Luis. Thank you, Harry.
Bye-bye.
Good to see you, Jane. Carlos.
Thank you.
Bye-bye.
Bye-bye.